siguiente ponente es Omar Ramírez Flores de la UABCS de México. La ponencia es Identifying Signals of Adaptative Selection in the Population of Fin Whales in the Eastern Pacific. Then,
Hello. So today I'm just going to be talking about my PhD project and about this beautiful whale. Just as a little bit of information, we know that since the human genome has been sequenced, there's been an increase in whole genome sequencing techniques. And this gives us the chance to actually look at the good positive selection analysis and try to determine the information in a complete new data set, right? So in order to do that, we can take two approaches. The first one, which is comparative genomics, which takes several species and compares their genomes and try to look for regions of genomic of positive selection. And the other one, which is the intraspecific one, which is population genomics. This one takes sequences, whole genome sequences from different individuals and compares them amongst each other. We're going to be focusing on this one. Now, a little bit about the fin whale, or Balaenoptera physalus, is the second largest animal in the world, reaching from 25 to 26 meters long. It's the fastest whale in the world. It reaches between 60 to 70 kilometers per hour. And we know that this is also related to the feeding strategy. So another important characteristic that is hypothesized to be influenced in their feeding is their coloration. As you can see, the right part of the rostrum is white and the left part of the rostrum is black. And we believe this it hasn't been proved, but we believe that this has an advantage in their feeding strategies. Now, so we have three populations of fin whales in the Eastern Pacific, one that goes from the Gulf of Alaska all the way to California, and one that is in the Gulf of California, which is a resident population. And this is based on genetics, acoustics, and photo-identification data. And one that is in the coast of Chile, which is believed to be a feeding ground. Now, each of these areas have different environmental factors, such as has been said by Daniela previously, the nutrient enrichment. So we know that the Gulf of California is a very nutrient-rich environment compared to the one from the Eastern Pacific. So, and in another case, the sea surface temperature, the Gulf of California is more hot than the one from the Gulf of Alaska and also from Chile, which can be seen here, but anyway. And this gives us the idea that given that they are subject to different environmental factors, we would like to know that they're probably subject to local adaptation processes. A recent paper done with for the demography of fin whales, especially in the Eastern North Pacific and the Gulf of California, indicated that there's a little bit of that there's a complete structure between the Gulf of California, which, as I said, is a resident population, compared to the Eastern North Pacific. Although there's a little bit of mixing, we can see that they're completely differentiated. And also, in terms of genetic diversity, we know that the Gulf of California is has a less diversity than the ones from the Eastern North Pacific, based on runs of homozygosity and the heterozygosity, means of heterozygosity. And we know that they diverged about 16,000 years ago during the last glaciation event that occurred in Illinois. And based on this, we know that the one from the Gulf of California and on the Eastern North Pacific remain their effective population sizes. And well, why would we try to do a positive selection study for fin whales, right? So based on just the information of neutral variation, we can define evaluationary significant units. But the actual definition of a ESEU also depends on the adaptive variation that occurs in a population or in a species. Now, as I said, we have the idea that there's processes of local adaptation to each of these environments, and given that there are different environmental constraints that can affect them, it's important to know how they have adapted to these habitats and how they have coped with these environments. So the research aims for this project is to first detect adaptive differences between the three populations of fin whales in the Eastern Pacific, and also identify outlier SNPs and do a genotype environmental analysis for each population to try to determine processes of local adaptation. This project has already sampled the 3 regions. We have 30 samples from the Eastern North Pacific, 20 samples from the Gulf of California, and 20 samples from the Eastern South Pacific, with the collaboration of different institutions. So how it's been done? We have the 3 main stages, which is the pre-processing, which is where I'm currently at, the mapping, and the population analysis. So the genomes, as I said, we have 70 genomes from fin whales, and the reference genome that we use is the blue whale for the mapping. This is because it has the best coverage that we have so far for all baleen whales, and also it has a very long scaffolds that helps us to actually do the mapping. And right now I'm in the pipeline review of the mapping for this process. Specifically about the mapping, we're using the software GATK. We are doing right now, this part, but as I explained, we have the alignment done for most of the 70 individuals. The individual variant calling has been done, and right now I'm in the joint calling process of these organisms. I'm still remaining to do the variant annotation, and once that's done, I'm gonna have my BCFs ready for the analysis. The population analysis can be divided in 2 sections. The first one, based on selection scans, we're gonna do selective sweeps, trying to look at soft and hard sweeps of selection, using 3 approaches: high frequency spectrum, extended haplotype homozygosity, and also the FST indices. This is just based on, we are also going to do a principal component analysis with pcadapt, trying to determine also processes of selection based not only on FST statistics. Once that's done, we're gonna do a GEA. We're gonna take the outlier SNPs and the environmental variables. Like I said, it was funny that Daniela mentioned it, but I'm also going to use sea surface temperature, chlorophyll, which is an indicator of nutrient enrichment, and also salinity, which has been indicated that are the main factors that influence the populations of marine organisms. And this is going to be done with 2 approaches, one which is redundancy analysis, which is a multivariate approach, and random forest, which is a machine learning algorithm. What we expect to obtain, hopefully in the next 2 months, is to actually suggest possible environmental significant units for these 3 populations, and also see if there have been processes of local adaptation for these whales, and also see how they have coped with their respective environments. And with nothing else, I'd just like to thank you, and if you have any questions, I'm open for them.
Thank you, Mar. Do we have any questions here in the auditorium? Yeah.
Thank you for the nice talk. I was wondering how much do they move, these 3 populations?
So this is one of the questions that I was afraid to get asked because much of the information that we have comes from the North Pacific. The South Pacific, we actually don't have too much information of satellite data about how they move. Like I said, we believe it's a feeding ground, but this is the area that they sample is a marine protected area, which they only sample in that region. So in the case of Chile or the South Pacific, we actually don't know if they move to other areas. However, we know that the ones from the Southern Hemisphere do not move up, and the ones from the Northern Hemisphere do not move down. Having said that, we know that the ones from the Gulf of California, the ones that stay there, that we identified that they are from that population, stay all the year around that area. They do not leave. They have migration inside the Gulf of California, but they do not move outside. We have seen the ones from the Eastern North Pacific move into the Gulf of California, which explains the admixture that happens between them. But that's just one case. The ones from the Eastern North Pacific, their migration movements are also not well understood. Like I said, they have 2 management stocks, one which is in Alaska and the one from California, but that's just because it's better for the management government there to work with those stocks. It's not based primarily on biology. They have some information, but that's just in the management details for that.
Thanks for the talk. I was wondering, what is the relationship between chlorophyll content and a possible selective force in these kind of organisms? Is
Yeah,
something-
so the main question is, how do I know that the SNPs are going to be correlated to chlorophyll analysis, right? So
no,
just-
like
no?
why would you choose chlorophyll content as a factor influencing
So
as a selective force in these
okay,
kind of animals?
okay. So chlorophyll levels are related to the nutrient enrichment, right? So nutrient enrichment indicates that there's phytoplankton in the area. Phytoplankton creates zooplankton, and zooplankton is what the whales feed. So what we know, for example, is that the fin whales that are in the Gulf of California, based on just their movements, they are dependent on the food availability. So in previous studies that are not just based on genetics, they sampled the whole Gulf of California, and based on movements of satellite tracks, they determined that when there was a increase of nutrient enrichment, said chlorophyll, the whales were around that area. So that's just explaining that when they are feeding based on chlorophyll, because obviously that creates the whole trophic movement of the species and how everything fits, that's just an indicator of how they actually move around the area and also how they implement. Now, that's just on movement. Like I said, the Gulf of California is one of the most nutrient enrichment areas in the whole world. And that's an explanation why the ones from the Gulf of California stay there and do not move outside. Because a resident population is not obliged to live in that area. They can move, but they decide not to. Why? And that's one of the questions. Probably there's a process of local adaptation. I cannot say that right now. Probably in the next 3 months, I can explain it to you that and say, yes, it occurs. But that's one of the ideas why we use chlorophyll.
Okay, thanks.
Yeah.
I mean, it was a leap for the ones that, for the people here that don't work with animals. So I just wanted to understand a little bit more. Thank you.
Okay, thank you, Mar, for a nice presentation. Then we have to move on.